could be function in either protein or lipid synthesis. And the detoxification potential function could be because smoothie R is involved in detox. Cell Z could be a number of things. It could be for transport, for things like water transport in plant cells or oxygen transport in animal cells. For example, your red blood cells, when they're developing, will have all of the cell parts. They'll make a crap ton of hemoglobin, and then they'll shoot out their nucleus and any other organelles they don't need, and they'll become wandering bags of hemoglobin. Water transport cells in the xylem in plants are dead cells that are pretty hollow, and they're just tubes there to transport water. They could be used for protection, so some of the cells like that produce nails, cells that produce cork and trees that don't really have anything except for structural proteins. For support, like those structural proteins in, like those structural cells in plants. Storage, so for example, hemoglobin stores oxygen and you don't need anything else in there, or they could be undergoing apoptosis. All right, we went over this when we went over, I think, the semester 1 exam, but I'll go over it with y'all. In an experiment, rats averaging 300 grams of body mass were tested several times over a 3-month period. For each individual rat, urine was collected over a 3-hour period after ingestion of 10 milliliters of liquid. One of those was water, one was 1% ethyl alcohol, one was 5% ethyl alcohol. The volume of urine was then measured and results were averaged for all individuals within each experimental group. The data are shown in the table below. Pose 1 scientific question that the researchers were most likely investigating. What was our independent variable? Well, not the water. The urine was what we measured. So our dependent variable is our urine volume.
And then the independent variable is the rat.
No.
Damn.
What else is changing in this experiment?
Oh, the is it the solution? I knew it.
So the independent variable is the concentration of alcohol. So, for any sort of scientific question, if you're having trouble framing it, you can always write it as, how does, and then you write your independent variable, affect, and then you write your dependent variable. So in this case, it would be, how does the concentration of alcohol affect the volume of urine produced?
There'd be more, because like it's trying to get alcohol out.
Well, you're going into more detail than we need. Right now, we're just going over what was their experimental question.
Will they pee more?
What does it look like in the data?
Thank you.
Mm-hmm.
So thank you.
How does the concentration of alcohol affect the
Funny. Sneeze.
volume
Just trying to make a print.
of urine produced?
Make it perfect. Make sense?
Mm-hmm. Can you see if you write on
Alright, so some of the appropriate questions are things like, how does alcohol consumption affect urine output? How does alcohol consumption affect regulation of the kidney? Our question also would earn points. B, state a hypothesis that could be tested to address the question you posed in part A. What is a good format for a hypothesis? Lester.
The if-then?
Alright, if-then. If then you describe how you manipulate the independent variable, and then you describe the change in the dependent variable you expect. So, if rats consume more concentrated alcohol solutions, what do we predict will happen?
Then they will have more urine out too.
Then they will produce more urine.
You're in a lot of trouble.
That's bad.
Urine what?
A lot of trouble.
He said you're in a lot of trouble.
You just drunk some alcohol.
Doo-doo-ch.
My tummy hurts.
Go Dookie.
Some kid in there just
Alright.
standing.
Remember, this will be online, that's why I'm rushing.
That's what I'm saying.
So, some of the hypotheses you could use are things like alcohol consumption increases urine output in rats,
You just stand
alcohol
there, you looked at
consumption
me
increases
when I wanted
water
to go.
retention,
Listen,
any sort of
Is that even using my
any
just stand
sort
there like that?
of viable hypothesis
Example.
would have earned you points. And by the way, this one was independent of part A,
show me
so even if you didn't earn points in part A, you could earn points in part B.
Damn.
Look.
Alright, so C, using the data in the table,
I'm talking
describe
to
the
dude.
effect of ethyl
Said you
alcohol
wanna
in
buy a
urine
sticker
production. How
I'm gonna
does
make my own.
ethyl alcohol
They
affect
got
urine
one.
production?
They got some girl with like a banana.
I'd rather make
Ethyl
my own.
alcohol increases
He's on the dollar. He ain't trying to buy it.
Ain't got no money, bro.
Urine?
the amount
Give me a shout out
of
on
urine
Instagram.
produced.
I don't have an Instagram.
What kind of life do you
You
live?
don't have Instagram.
When you guys were
Where you
doing
at?
this on the test, a lot of you guys put things like, oh, ethyl alcohol changes the content of the urine. But where up there do you see any measurement of what is in the urine? Do they tell you what is in the urine?
No.
What is the only detail they give you about the urine?
How much urine is produced?
How much there is. So that's the only thing you can actually draw a conclusion about. So that gives you another point. Alcohol consumption increases your urine output. And last, but certainly not least, our 8th question. The figure above represents a generalized hormone signaling pathway. Briefly explain the role of each numbered step in regulating target gene expression. So what is step 1? What's happening there?
Entering the plasma membrane.
That
Is
is
anything going into the plasma membrane?
yes.
So this thing is moving straight through?
No, it's trying.
It's not trying to. It's interacting with something in the plasma membrane. What do we call this thing in the plasma membrane? A receptor.
Receptor.
It receives information. This guy is a ligand or ligand. What are all of these guys in step 2? A generalized term for them. Let's say that the ligand is the first messenger in a pathway.
Those are the other messengers?
They're the second messengers.
You look sleepy.
And what is this guy in step 3?
I like it.
Probably not.
Helicase.
It's something that's affecting gene expression. What are some molecules we learned about that affect expression of genes?
Enzyme?
Protein.
Transcription factors.
Oh,
It's so hard because life is a very complex, multifaceted mystery.
well, you can just get a gun and shoot yourself in the mouth and it'll be over.
If you want simple life, go be a bacterium.
Oh my God.
I'll be a fungus just to infect someone's
That's
with
why I
some
want to
feet.
live in the cabin.
That still won't make you a bacterium, sweetheart.
I'll be a fungus and infect people's feet.
Who said I
life
wanted to follow you?
Right, Michael wants to be athlete's foot when he grows up. Alright, so what's happening in step 1?
You're a racist.
Partial receptor.
Maria.
Oh, I
The
know that.
ligand
I know that girl.
binds to the receptor.
Boy. Why these niggas fucking fighting?
He said he wants Maria's
That's what he says
Instagram.
on the video.
Boy.
Nah, but he said nice stuff.
Ask
And
anybody. Ask anybody.
It's from
Maria.
we get to each other.
Her homegirl. I
and
said ask anybody.
starts
He
the
said,
signaling.
I don't want her Instagram. I just want a shout out, bro.
You one of them guys that ask for shout outs?
The girl,
You
she,
can give your own shout out on the video.
the girl that, the
That's
girl
the
that,
ball.
I got
Shout
more,
your, look, shout
I
yourself
got more
out right
followers than the girl she just showed me.
now. Shout your Instagram out right now. Dang, miss it went
Don't
for,
you do it!
Hey, miss it went for 43 minutes.
Yeah.
Hey, shout mine out. I only got 2 followers and
Hey!
8 accounts.
Quit trying to use my videos to, like,
I don't think they're gonna follow me anyway. They're all nerds.
okay, what's happening during step 2?
I'm
They
just
are
winning
reading the
these
message.
push-ups.
They're gonna get better.
Second
They're
messengers.
better at it, you
You coming up too slow.
know? Were you gonna show?
Are
You gonna show?
passing the message
Yeah,
along.
nigga, push this ain't nothing, boy. How many you want, boy? Oh, man, it's 5. If you get like a 6.
15. 15.
What does Lester have?
15 what?
Push-ups.
All right, and what's happening in step 3?
I'm doing Lester's Instagram. Let me see.
So step 3, transcription factors.
Recording being beginning.
Them the little.
That have been activated by the second
Why do
messengers.
people taking
Those are
over your
joke
paid walk?
I swear.
Let me see.
So, guys, transcription factors activated by second messengers do what? Well, they're gonna encourage transcription, so they're going to activate gene expression.
Let's
Because they're not DNA or RNA polymerase. They're not actually making
Yeah,
DNA or
swimming
RNA.
hole.
What they are doing is making it easier for RNA polymerase to bind and start producing that gene. So they're going to activate
Rule number
gene
1.
expression.
Number 1. You need more clothes.
I have friends that have clothes that can do that.
What are you talking
And then if we look
I'm
at the
offended with
scoring
that.
guide, you'll be able to see. So step 1, we have something
Number 1,
binding
you
to a
need more
receptor
clothes.
and it initiates or starts out the
To
signaling.
jump in.
Step
Full
2, you
sweatsuit.
can say it's something like an
And a helmet.
intracellular cascade. You can say you're amplifying the response, transferring it to the nucleus, etc. And step
Do you
3,
have
you're
shoes
regulating your
on?
gene expression, where you're either going to turn off or turn on a particular gene. Can you guys turn off the video?
Scumbags.